Hello, everyone. Let's see. I'm going to start my webcam. Here we go. Hey, everybody. As you can see, I'm in my wood shop outside because my family is asleep, and I'm recording out here because we're all kind of, or well, luckily I'm not, but they're kind of sick, and but they're doing good. They're getting better, and so I'm not going to be able to be at school this week. um, Wednesday, Thursday, Friday. But hopefully, if everything goes as planned, I'll be back Monday. So, but anyway, that's the plan for me. But what I thought we'd do is just use technology, use YouTube to be able to record what we're going to do, and then I'll post it on YouTube, and then the substitute, Miss Schultz, will show it to y'all Wednesday, Thursday, Friday. So I'm going to record this video today, and hopefully it won't be very long, maybe like 20, 25 minutes, and you'll be able to take notes as I speak through the notes. And obviously, you don't have the handouts yet. And I thought about trying to get all that printed out, but since I can't come up to the school and it's a lot to print out, you guys know how big those packets are. I just wasn't going to do that. So, but I know Coach Harris did get a couple sheets printed off for me, so I think we may have give you those as some work to practice on some of the things that we're going to talk through. But the main thing will be the way the class will go is Miss Schultz will pull up the YouTube video, and she'll play it as a class. And what I need you guys to do is pay attention. Just pay attention, and there may be times I'll probably go faster than you can take notes on the video. Um, but you might, if at times, if you're taking notes, you could ask Miss Schultz politely to pause it, and she could probably pause it for a second to do that. But remember, you have this on YouTube. You could always go back at a later date and watch the video. But I don't see a reason why it couldn't necessarily be paused for a second. Um, you could catch up with what a slide said, and because I'm going to post this screencastify right into YouTube, and so you could go right on there and watch it. So I think it will work really well. I did a little practice one, and it seemed to work well. Okay, so I really want to be there with you guys. I don't like this way, I think you guys know that, but it is what it is. So let's make the most of it. And if you guys will use this, we'll refresh when I get back on the next Monday, and this will really keep us going in the right direction. So anyway, let's get started. I guess with we'll start with what we're going to talk about is genetics. We talked about heredity, DNA, the background for this, what we're talking about right now. And I think as you'll see when we go through some of this stuff, a lot of the same terminology is going to come back up. And hopefully it'll make it where it's not as foreign to you. And you may have to refresh a little bit, but that's what I'm for. And we'll do that. Okay, so here we go. Let's see if I can. All right. Okay. All right, so we should be good. All right, so Mendelian genetics, and you've probably heard of Gregor Mendel before. But if I don't know if you didn't know, but Gregor was actually not his name when he was born. He was born Johann Mendel. Gregor Mendel is a really neat dude. We'll talk about him a little bit. Okay, let's put this up here. Whoops. All right, so there. Okay. So it's his background. With the exception of gametes, aka egg and sperm, all cells in your body are diploid. Two copies, diploid, think die, like a die, you got two dice, right? And contain two copies of each chromosome. We get one from mom, one from dad. As it says there, one copy from mom in her egg cell and one copy from dad in sperm cell. And then it says how many total chromosomes make up who you are. And we've talked about that number before. And you may know that you can kind of say the number is two different ways. So some of you might know, and you might say it right now in class. But without further ado, that number is 46. We said the number is 46 total, or you also may say and accurately say 23 pairs of chromosomes. So you get 23 chromosomes from mom and 23 chromosomes from dad, right? Which you could say is 46 total chromosomes. But you get, like I said, two of each one. So there's 23 different ones, right, that are very different, but you have 2 copies of each one. And so chromosome 1, you like have mom's chromosome 1, right, and dad's chromosome 1. And so if you count them as this is chromosome 1, this is chromosome 1. They're a little bit different though, because, and we're going to get into this term, is they contain different copies of a gene. We're going to get into what you call that in just a little bit. But they're still chromosome 1. So if you count them as his 1, this is 2. That's 2 chromosomes, but they're both chromosome 1, right? So it's a pair. So I like to say 23 pairs. That kind of keeps me in line, but you'll often hear 46 as well. How many total chromosomes make you up who you are? 46 total, or 23 pairs. Yeah. There it goes. Okay. All right. Go ahead and put all that's up here. And guys, this is a slide where it'll probably take longer. It's but may contain. It'll probably I'll go through it probably quicker than you can write it. But again, this may be one that you can pause. Okay, because this is some important terminology on this slide. And if you'll notice, uh, this first term is review. The second term is totally new. Um, the bottom term shouldn't be totally new, but it may not be something you're super familiar with. Okay, but that homo prefix, we've discussed it a lot. The homologous chromosomes, you probably have an idea of what that means. Okay, let's go through the first thing. Background. Gene. A gene is not the genes you're wearing right now. A gene we're talking about, right, is a section of DNA that is made into a protein. Remember, you are flesh and blood. What we see when we look at you are your proteins. So that is so key. We've talked about that so much, and I hope if you don't leave this biology class without understanding the process of getting from DNA to protein, right, and how that happens. And then that we talked a lot about when you go from DNA, we have to transcribe, which makes messenger RNA, right, which then can leave the nucleus because there's no ribosomes in the nucleus, go into the cytoplasm or rough ER and find that ribosome and translate the messenger RNA into a polypeptide, which is eventually folded up into a protein. Right? Okay. It says there, here, since a gene is a piece of a chromosome and we have 2 of every chromosome, we have 2 copies of instructions to make every protein in our bodies. Bam. That's so important to realize, is that's why we have 2 chromosomes, right? We have one from mom, one from dad. So you may have on chromosome 1 from mom, it may mean you're going to have brown hair. On chromosome 1 from dad, it may mean you're going to have uh, red hair. Okay, so and just for simplicity's sake, well, because this is the first thing we'll talk about. Let's just say it's all about dominant and recessive. It's not, right? But let's just say it is. Let's say red's dominant to brown. So you have the brown gene, right, for hair color, but you also have the red gene for hair color. And because red is dominant, that means you will have what you will see, right, when somebody looks at you is red hair. But in there, you have the gene for brown hair. So that's a kind of a neat thing that you could possibly have a child who has brown hair, depending on what the other person in the relationship has. Okay. Uh, so alleles. So I always like to think about an allele as like a type of ice cream. So it's all ice cream, but you have a lot of different flavors of ice cream, right? You can have chocolate ice cream, vanilla ice cream, all those different types of ice cream. Way more than that, right? And that's what it says. An allele is a different version of a gene, right? Brown hair, that's one version of a gene. Blonde hair, that's another version of a gene. Uh, Red hair, that's another version of a gene, of the same gene. They're all the same gene, it's different versions of it. And what do we call that? We call it alleles. An allele, Alleles. Okay. Um, all right. So and then it gives you the example down there of what I was talking about. And homo means the same. So homologous chromosomes are the matching chromosomes from our mom and dad. Right? They contain the same genes in the same locations, but may contain different alleles. Right? So you got chromosome 1, chromosome 1. Well, at this crease on my finger, is the gene. Look, I mashed my finger the other day. That's kind of ugly looking. Sorry. At this crease on my finger is the gene for detached earlobes. Well, at the same location on the other chromosome is going to be the gene for detached earlobes. But this one may be attached. Now, I'm just making this case up. I don't actually know which one's dominant or recessive. But let's just say detached is dominant for attached. So they're in the same location. They're homologous chromosomes. They have the same gene, same location. Okay. All right. Here we go. Let's go to the next slide. All right. Very quick thing on Mr. Gregor Mendel. Super interesting guy. He made very humble beginnings. If you don't know about Gregor Mendel, I hope you'll know a little bit about him after this. So just from the standpoint, most people just know he's this monk, right? Austrian monk who came up with genetics. And that's probably, I guess that is what he's most known for. But he came from a super humble beginnings. I think he was born in 1822. And he was born on a farm to a farming family. No, he wasn't like this well-to-do person who had everything to start off with, and but no, his very very humble beginnings, and but his teacher noticed that he was very gifted in school, and I think it was at the age of 11 he entered into a program kind of to I think it was a specific program, maybe I may be off on this a little bit, but for math and science, and kind of from there he ended up going on his journey to basically making some of the biggest discoveries in science history, and and is known now as the father of modern genetics, the father of genetics, and it all started with these pea plants, and being able to very tediously, very tediously. Crossbreed and breed these pea plants to find out some really interesting things. And he came up with these 3 laws of inheritance. And it's called the law of dominance, the law of segregation, and the law of independent assortment. And we'll go through all 3 of those. Okay, so what did Mendel do? Here you go. This. I have to figure out a way to. Yeah. All right, so it says in his experiments, crossing pea plants, he made sure that he had control over breeding. Mendel used pea plants that typically self-pollinate. And so if you look at there's some, let's go over this right here real quick. You see how you might be able to see this. This says P generation. That means parent. So like the original ones, the parents. And then this F1 is the first generation after the parents. F2 is the second generation after the parents. And so, but let's go back over to here. Number 1, he had control over breeding. Okay, very important. He had to make sure what he thought was happening really was happening, right? That was super important. He used only pure breeding plants. That meant that, like a pure breeding plant, it just means that if it's a purple plant, if you breed it, it's only ever going to give you purple plants. Okay, if you breed a purple plant with a purple plant, you'll always get purple plants. And this one, if you breed a white plant to a white plant, you'll always only get white plants if they're pure breeding plants. Okay, that's what he was saying there with pure breeding plants. It's uniform, genetically uniform. Okay, and then number 3, it says he observed either or traits. Choose pea shape, pea color, pod shape, pod color, pod height, flower color. Okay, either or, this or that. So it's very easy to tell the difference, okay, between them. So this is really, let's just go over this picture right here real quick. This is what happened in a nutshell. So Mendel took purple flowers, white flowers, bred them together. And he noticed that all he got in the next generation was purple. All he got was purple. And it's like, well, what happened to the white? What happened? Was it gone forever? Was it gone for good? Did he get rid of this white gene by crossbreeding purple and white? Because in the F1 generation, every flower was purple. Well, he had something really smart. He said, well, let me take 2 of these all purple flowers that came from the cross of purple and white and breed them together. And what did he find? is the white came back. And it came back in a really specific ratio. It was 3-1. So for every 3 purple flowers he got 1 white flower. And that was a big deal. And then he figured out some more from that. But that was the ratio, 3-1. 3-1. So the white flower was not gone. It was hidden in this purple flower. Okay. All right. Okay. So this is what we were saying. What is a cross? This is kind of what I just described in that picture. It's the mating of 2 organisms, and this is says it here. What I just said on the last slide, and you want to write that down. This here, write that down. That the P is parental generation, F1 is first generation, F2 is second generation. That's what we were saying. And if you notice, here's probably a better picture of it. You can see the parents, all purple, mated with all white, and you get all purple. Like, what in the world? That's kind of cool. And then if you breed the two of the purples together, you get this, like we said, 3-1 ratio of purple to white. Okay. All right, the significance of alleles. All right, the significance of alleles. So this is terminology, guys. We've got to be able to communicate with one another. So this stuff's actually really important that you understand how we write things. It says alleles, how are these different versions of a gene, brown hair, blonde hair, how are they going to be communicated? Well, we use uppercase for the dominant version of the gene, the dominant allele, and we use lowercase for the what we call recessive allele. Okay? So it says, remember, each individual has 2 alleles, one from each parent, one from each parent for every gene. Okay, so you have like, what is it, like 30,000 genes or something? I may be off on that, something like that. So, and that's between all 26 pairs of chromosomes. Okay, so homologous, homo means same, hetero means different. So homologous is 2 of the same alleles. For example, like big A, or little a. So that's if this was your genotype, big A, this was called a genotype, big A, or little a. What that would mean is that you would be homozygous. Homo means the same, zygous for a zygote, but so the same 2 versions of the gene. Okay, got the same one from mom, same one from dad. Or you could be heterozygous. Hetero means different, so it's 2 different alleles, big A, little a. You got a different one. You got one from mom and a different version of the gene from dad. So say. Okay? Okay. And this is what we saw in Mendel's pea plant. Okay, the law of dominance. Okay, so again, this may be one where you have to pause it a little bit, but okay, it says, the law of dominance says, a dominant or strong, it's not really stronger, it's just a good way to think about it, I guess. A dominant or strong allele will express itself over a recessive or weak allele. It doesn't necessarily mean that the dominant allele, like I said, is actually better. That's not the case at all. Or the recessive allele is worse. That's not the case at all. It's just what we call them so we can communicate. So you know what I'm talking about. I know what you're talking about when you say dominant and recessive, or strong and weak. So dominant, if present, will always express the trait. So it'll be seen. So that means if red hair is dominant over brown hair, let's just read what it says there. If brown hair is dominant over blonde hair, and big B represents the allele for blonde hair, and B represents the allele for brown hair, if you inherited the dominant allele, which is big B, from both of your parents, you will have brown hair. Okay? And it says whether you are big B, or big B, little b. Okay? Either way, you're going to have brown hair. So there's a much more likelihood, it's actually 75% chance, that when you cross, if you're heterozygous, that you're going to have the dominant allele trait. So it's only 25% chance if you're heterozygous that you'll have if your parents, I should say, are heterozygous, that you'll have the recessive trait. And we'll see that in a little bit. So recessive allele will only have the trait expressed seen when the dominant allele is not present. So, for example, little b, if you had both recessive alleles, you have to have both to have blonde hair. You have to have a recessive from your mom and a recessive from your dad. Okay? All right, genotype versus phenotype. Okay, guys, this is, again, some important vocabulary words. Genotype versus phenotype. Oops. And again, this is just vocabulary words. Genotypes are letters. Phenotypes are physical traits. That's the way I think about it. Genotypes are letters, like big B, big B, big B, little b. Okay, so it says they're genes that code for flower color, such as big F, big F, big F, little f, or little f, little f. Okay, those are genotypes. They're letters. Okay, they're the actual alleles. Okay, the versions of the gene. I'll say that again. Allele. It's like, hey, what flavor ice cream do you like? You'd say what? You could say, what allele of ice cream do you like? That would be the same thing. Science joke, right? Hey, what allele of ice cream do you want for dessert? And you may say vanilla. Another person may say chocolate. Right? And then you'd be, see, it's just a different version of ice cream. So it's all ice cream. Okay. So that's if you can get that, that's what allele means. If we can get that, we'll be on the. It'll help us understand what we're talking about. Okay, phenotype, the physical traits, characteristics seen in an organism. So what does it actually look like with your eyes, right? Physical trait. When you see it, what does it look like? That's a phenotype. Okay, that's the physical trait is the phenotype. Okay. All right, law of segregation is Mendel's first law. Oops. Okay, law of segregation. It says that when chromosomes separate in meiosis, and guys, this is going to be maybe a little bit tough, is because I realize that we didn't get meiosis. We actually have to go back and get meiosis from the last unit. And we're going to do that. So if this doesn't look, we've already done mitosis, but we didn't get meiosis. So we're going to go back and get it. So don't worry, but I still think everything we're doing here you can understand. So, but yeah. So meiosis is really just mitosis twice. Like this would be the end of mitosis here, but meiosis does another division. It does it one more time. Okay. All right. All right. So it says, for example, if a man has so the law of segregation says, let's read it. When chromosomes separate in meiosis, each gamete, egg or sperm, will receive only one chromosome from each pair. Okay. So remember, let's look. What are we looking at? See if we can get this here. And that's kind of what we can get from this. Okay, we're looking at a cell. Okay, in this case, we're looking at a spermatocyte, which is where sperm cells come from, or we're looking at an oocyte, which is where egg cells come from. Okay, so um, so in this case, we have duplicated chromosomes, right? S phase has happened, right? And we're about to start, in this case, not mitosis, but meiosis. Okay. So what we see here is, let's just say here, we have chromosome 1 for mom, chromosome 1 for mom, right? Because these two are called sister chromatids, right? Hope y'all can see what I'm looking at. This blue chromosome right here, I think you can see my cursor. This will work. Let's do this. Aha. See that? That one. Yeah. There we go. I forgot about that. That is a sister chromatid. Okay. That's a sister. Okay. Well, these two things are sister chromatids. They're sister chromatids. All right. We'd call these. I'm gonna put S. Man, that's easy to write. Shoot. Okay. I'm not gonna do that much more. Sister chromatids, and we call these two together homologous chromosomes. Okay. So these would be sister chromatids. These would be sister chromatids, but these two would be homologous chromosomes. Okay. So chromosome 1 for mom duplicated, chromosome 1 for mom duplicated, chromosome what should I say? Chromosome 1 from dad duplicated, chromosome 1 from dad duplicated. Okay. All right. And then if you'll notice now, what we separate in meiosis is we separate meiosis. We call it meiosis 1. Is we separate homologous chromosomes, not sister chromatids, and then you separate down here. You'd separate sister chromatids. Okay, so again, that's meiosis. What I just described there to you was meiosis. So hopefully we're going to go back and get that. Okay, but again, we can get that. The main thing I want you to get from this right now is what this box says. What does the law of segregation say? When chromosomes separate in meiosis, each gamete, egg or sperm, will receive only one chromosome from each pair. And if you notice that, look, it did. Down here, you got one from here, okay, and one from here, okay. There we go. Okay, so all this separated into this. Go back to this. There we go. Okay. Oh no, let's erase that. Yeah, I'm gonna try not to draw anymore. Let's see. If I need to, I will, but if I might not. Okay. All right. So law of independent assortment. This is another one, right? Another law, Mendel's law. The assortment of chromosomes for one trait doesn't affect the assortment of chromosomes for another trait. So basically what we're saying here is that, so let's read this here. This allows for any combination of maternal and paternal, mom and dad, chromosomes to be passed on because homologous chromosomes line up randomly during metaphase. Okay, remember metaphase line up at the middle, right? Okay, so and remember, think about it. Sperm cells, there's millions of them, right? So you get millions of different combinations possible for any offspring. That's why every human's unique. Okay. All right. So law of independent assortment. And that may, if you didn't have time to write that down, this is definitely something you want to write down. You have questions about what is law of independent assortment? What is the law of segregation on a test? So I'm going to pause that. I'm going to try to finish this up for you guys now. Okay. And so this is something that I want you to get here. This is where this is the Punnett squares. I think you did this in middle school, in 8th grade or maybe 7th grade, somewhere around in there. You did this, so you're kind of doing it again. So what is a Punnett square? A diagram that shows the probability of inheriting traits from parents with certain genes. It's probability. Like, what's the probability when I flip this quarter, it will land on heads, right? 50-50. Kind of the same thing here. It's just probability. Okay. So a monohybrid cross is a cross between two organisms looking at one trait. Okay. Possible genotypes of offspring, okay, are in here. So we would have put parent number 1, they're heterozygous, big A, little a. Parent number 2, big A, little a. Then I add them in. Move this A here, that A there. This A here, that A here. Here, or well, here. It doesn't matter technically. The convention is that the big letter goes first. Okay, so you put the big letter first, put the little letter second. But it actually wouldn't matter necessarily if you put little a, big A. The big A is dominant. So there you go. So in this case, it doesn't say what this trait's coding for, but you have homozygous, the same, and it's dominant. Heterozygous, homozygous for the same, and then it'd be called recessive because they're both little. Okay. All right, I'm going to do one practice or a couple practices with you, and then we're going to be done for today. And then I think you actually have a practice worksheet to do with this, and we're going to be done with this video. All right, so let's do one together. Actually, we'll get the marker out and see if I can do this a little better. All right, here we go. All right. In pea plants, it says round seeds are dominant to wrinkled seeds. All right, so let's just use R for round and wrinkled, okay? And it tells us round seeds are dominant to wrinkled seeds. So big R is going to be what? Right? Big R will be round. R-O-U-N-D. I'm writing with my mouse. This is not easy. Okay? It'll be round. And of course, little r then would be, I'm just going to put a W. for wrinkled. Okay, so when we put them over here, it says cross. I'm going to put them on the Punnett Square. It says cross a homozygous recessive plant with a homozygous dominant plant. All right, so homozygous, it doesn't matter what side you put them on. So homozygous recessive, if it's recessive, we know it's going to be it. And this homo means the same. So little r, all right? And it says with a homozygous dominant plant, big R, okay? All right, now I'll fill them in. Well, you look here, big R, little r. This is Mendel's cross, guys. This is what he did. And if you'll notice, this is exactly what Mendel did. And in this, it would seemingly show you that the wrinkled plant is gone. You get no more wrinkled seeds, right? Because all you're going to get when you do this cross is a whole bunch of round seeds, no wrinkle, okay? But then you say, well, cross two heterozygous plants. Well, these are heterozygous, right? So let's just say we take this one and this one. Bring it right here, big R, little r, right there. So that's that one. I'm going to take this one, big R, little r. All right. All right, so round again, round again. Oh, but wait, there it is. Little r, little r. It came back. Now we have wrinkled back, okay? Now we have wrinkled, so it showed back up. It showed back up. So there it is. Okay, so it's not gone. 3-to-1 though. So if you have 400 plants, okay, 100 of them will be, 300 of them will be round peas, and 100 of them then will be wrinkled peas. Okay. All right. So it says cross heterozygous plant with a plant that has wrinkled seeds. All right, heterozygous plant, big R, little r. We know that. It says heterozygous, bam, one big, one little. Okay. Allele. One dominant, one recessive. I should say that. One dominant, one recessive. With a wrinkled seed. So we know wrinkled. If it's going to be wrinkled, what's the only choice for its genotype? Remember, the letters are genotype. It's the only choice. Little. Yeah, little. All right, let's do it. Big R, little r, big R, little r, little r, little r, oof, little r, little r. And now it's 50-50. So in this generation, we'd get, if you have 400 plants, you'd have 2 of them that are 200 seeds that are round and 200 that are wrinkled. Okay? Awesome. That's that. Okay? That's a lot easier. Okay. So, guys, we're going to stop there. Dihybrid cross. Oh, man. So we're not going to do a dihybrid cross yet. We'll stay with monohybrid cross. And like I said, I think you have a worksheet on this that Ms. Schultz will give you. So thank you, guys. I hope this was beneficial to you. I hope this will keep us on track. And I'll try to make another one of these tomorrow and let you guys watch the next one tomorrow. Like I said, I hope I'll be back on Monday is the plan. And we'll get back going. Guys, please be on your best behavior. I want that so much. And I hope I said that beforehand. That's what I expect. And I know all of you can do that. And I miss being there with you guys. Ready to get back going with this next semester. We're ready to rock and roll, right? And have a really great second half of the year. Archery's about to get going. It's about to be a fun second half of the year. All right, guys. Anyway, see y'all. Everything goes well on Monday.